All right. And our last speaker before the lunch presentation is Dr. Corey Brizzee. I hope I'm pronouncing your last name correctly. And he's a strain engineering scientist in yeast and protein engineering at Demeetra. He has a PhD in molecular and cellular biochemistry from the University of Kentucky. Corey is now leading a team at Demeetra to enhance its proprietary Cas-CLOVER RNA-guided nuclease for ultra-high efficiency and specificity knockouts and knock-ins in a variety of cell types for synthetic biotechnology and bioprocessing. Corey, welcome.
Thank you for that introduction. So today I just wanted to give like a brief overview of Demeetra AgBio's Cas-CLOVER technology, which has been validated as the proven alternative to CRISPR-Cas9. So how our technology works is we use a catically inactive Cas protein as an RNA-guided fusion protein, and this is fused to an endonuclease called CLO51 from a Clostridium bacteria. Now, CLO51 is an obligate heterodimer, so it needs to dimerize for cutting. This results in large deletions with 4 base overhangs. And the 4 base overhangs creates a higher knock-in efficiency during cutting. So this works, our technology works seamlessly with the existing guide RNA infrastructure that you would typically use for CRISPR-Cas. We use a guide RNA pair, which creates a higher fidelity and higher efficiency system. Our CLO51 dimerization also has a very flexible spacer length, and this creates a user-friendly experience when you're actually designing guides for your system. A convenient method to screen for enhanced cutting efficiency, Demeetra uses a yeast add-to reporter assay. So Cas-CLOVER will do a double-stranded DNA cut in the add-to ORF, and then upon homologous recombination with a donor DNA fragment with flanking homologous fragments, it creates a full add-to gene deletion. This accumulates an adenine biosynthetic precursor and then results in a red colony phenotype. And then on the right side of the slide, you can see a typical plate from a wild-type Cas-CLOVER experiment. Now, a really hard thing that we've seen at Demeetra is this balance between cutting efficiency and cellular toxicity. A lot of times when we use wild-type Cas-CLOVER or wild-type Cas9, we'll see moderate cutting efficiencies, but you're also getting moderate cellular toxicity. So you don't get a lot of colonies forming on the plates, and if they are red, there's a very select few that are on the selection plate. And this can see on the other side of things where we look at inactive Cas-CLOVER or just putting a single guide expression only in our Cas-CLOVER system, you see a complete lawn of colonies, but there's zero cutting, which there shouldn't be. So we really wanted to find a balance between cutting efficiency and cellular toxicity, and we wanted the best of both worlds. Can we make a system that is over 90% cutting efficient and reduces cellular toxicity to a very low level? So we turned to a lot of previous literature on a similar endonuclease called FOC1. This is another type 2 restriction endonuclease, and this is often associated with zinc fingers. Now, CLO51 shares about 50% identity as well as 75% similarity between FOC1. And a lot of the variants that were made were looking at the ratio of on to off target frequencies without compromising the actual on target frequency. So these mutations were done via a structural model, and they looked at these mutations and these positions for these variants, and it was kind of spread where the DNA would be involved with for cleavage kinetics. So we chose the top 6 mutations that are very similar or equivalent to the FOC1 mutations. And we tested these out in our add-to reporter system, and this completely abolished almost all of our Cas-CLOVER cutting efficiencies, which just goes to show you they might be similar in amino acids, but they might act very differently as a complete protein. So we only had about one of these variants that really, they didn't help. The F44S mutation didn't increase cutting efficiency, nor did it decrease cellular toxicity. So we chose another approach looking at a structure-guided mutagenesis approach. So we did a Chimera X model, and this uses an AlphaFold 2 prediction software, which is the new craze for protein structure prediction. And we align this to the protein structure of FOC1. And I noticed that there was a very unstructured loop between the catalytic site and where the DNA is projected to bind to FOC1 or CLO51. So doing mutagenesis on this specific position, we had 3 variants that really showed us accelerated or higher cutting efficiencies with moderate lowering of cellular toxicity. So we had variant 1, 2, and 3, which had probably about 40% increase in cutting efficiency, and we were exceeding that 85% that we really wanted to get as close to 100. Everybody wants that. And we were seeing a lower cellular toxicity as well. So we took this to another level and basically did a very extensive screen to identify any type of variant that could get us, the best of both worlds. So a lot of these variants increased the cutting efficiency of our Cas-CLOVER system, but they didn't really decrease the cellular toxicity. There was only one that really popped out, and that was our variant number 14. So the typical plate would be, on the top right, and that would be our wild type Cas-CLOVER, where we basically started from scratch. And then our variant number 14, we see that full plate that's full of colonies, and indeed these colonies are all red. So we have almost 99.5% cutting efficiency with very, very low toxicity. So we've used this type of Cas-CLOVER system to create bioprocessing platforms for different customers and clients. This one is a collaboration with Dr. Molly Duman-Scheel at the University of Indiana. And this is making female-specific larvicidal IRPs. So we use Cas-CLOVER to create the bioprocessing platform, and we use that in conjunction with our super piggyback transposon system. And this is really good for being able to select for integration of a certain biopesticide. So as I mentioned, these are female-specific IRPs. We made yeast strains with varying levels of expression of these IRPs. And in a female or in a larvicidal killing assay, we can see all of our yeast strains when fed to mosquito larvae, female-specific larvicide killing happens, and it doesn't really affect the males, which is really good because females are the ones that bite, and no one likes biting mosquitoes. And they transmit diseases, and commonly it's West Nile and varying encephalitis diseases. And this can be validated with doing in situ hybridization. You can see targeted silencing of that specific target in these larvicidal brains. And we also have used our Cas-CLOVER technology to make GS knockout suspension cells, which is commercially valuable. We actually multiplexed this system using two guide pairs, and this created 196 base pair deletion in an exon, and then single cell clones were identified. We chose clone G4 because it had 100% viability with glutamine, and without glutamine it had 0 viability. And then the viability is recovered when you transform back in a construct with glutamine synthase. So just an overview of our Cas-CLOVER technology compared to CRISPR-Cas9. We have very low off-target effects compared to CRISPR. Both technologies have high activity. Our indel sizes are larger because of our flexible guide construction. We have high knock-in efficiencies because the overhangs are different. And our IP status is that we have commercial freedom to operate with a single simple license, whereas CRISPR-Cas9, you're probably going to have 2 or 3 separate, and you have pretty much major entanglements with the licensing. We offer a lot of bioprocessing evaluations, and if you want to learn more, just to keep time simple, just please visit us at booth 501. Thank you.
Thank you, Dr. Brizzee. So because of time constraints, let's take your offer to meet you at your booth.